Hi, good afternoon. My name is Pieter van Velen, and I will be your host today for this Wetsus webinar on applied genomics. With me here is Ines Dinkla across the table. She's a scientific program manager at Wetsus, and she's also the theme coordinator of the research theme Genomics-based water quality monitoring. And Ines will today monitor the chat and collect and synthesize your questions that you may have about the different presentations or in general about applied genomics or how you could apply genomics to your system or process that you are working on. The goal for today's webinar, is that we identify the difficulties and challenges of applying genomics to answer questions or to come up with solutions for real-world problems in the biological systems that you either study or work on professionally. In this next slide, I have tried to conceptualize the journey that we're taking today. On the left-hand side, you can see the question that comes up, either in water or other systems, about the biology. And if you try to understand the biology, you can either look at animals or plants that you can see, but there is this whole world of unseen biology that might be important to your system. With genomics, you can access that kind of information. And to be able to understand DNA or RNA-based information, we need reference databases that tell us which DNA belongs to which animals. And reference databases are important in that respect. Morten is our first speaker, and he will talk about reference databases and sustainable use of those and building them for future application. Then later on, I will take over and showcase some of the work of my colleagues at Wetsus to give some very broad examples in water and outside of the water scene on how we could use that genomics data to understand our biological systems and to discover bioindicators that can later be used in the applied field to monitor systems in an easy, accessible, and fast way. And that's basically the field that Aleida Holmes is going to talk about. She's sitting next to me. She works at Orvion, and at Orvion she has a long-standing reputation of working on in the applied field and applied genomics to tackle real-world problems. But the part of the title here is that we want to apply genomics towards routine use in water monitoring. And for routine use, we need standards, and we need a formalized way of how to deal with DNA, with genomic data, to answer questions in a standardized way. And here today we have Adri Atsma, and he will tell us from his experience either at a drinking water processing center at Vitens, his experience there in the lab, from analyzing their water on a daily basis, but also he's a delegate at the International Standardization Organization. He's the Dutch delegate there, so he has a very good insight in what is necessary for standardization. So I'm very pleased to announce our first speaker for today. That will be Morten Duholm. Morten is an associate professor at the University of Aalborg in Denmark. He will be joining us digitally from Denmark, I think. His department has a long-standing reputation in the microbial ecology of wastewater treatment plants. And they have uncovered a lot of drivers of microbial community dynamics in these systems, but also mapped global diversity, similarities, and differences among wastewater treatment plants. He will tell us today something about reference database and an important new reference database that he has recently published as Midas 4. So with this, Morten, I would like to give you the floor.
So thank you. So can you see my slides? Perfect. So, as you said, I'm an associate professor from Aalborg University, and I've been working with this Midas database here for the last 5 years, trying to improve that. So if we look into research recovery facilities like wastewater treatment plants and biogas production plants, we know that it's the bacteria in there in the archaea who's actually doing the job, so metabolizing all the substances and turning it into clean water or biogas in the end. So we need to understand all of these organisms. And if we take a microscope and look into the system, we can see that there are thousands of different species in there, probably even billions. So it's really difficult to understand these bacteria, and we need to understand what the different bacteria do to be able to steer them to do the correct job and avoid problems in our systems. So the traditional way to do that would be to try to isolate these different bacteria and study them. But the problem is that you can't really do that. There's a lot of bacteria you can't culture using different normal techniques. So instead, what we do is we turn our attention to DNA-based methods. So if we want to identify microbes in a system based on DNA-based methods, we in theory take a sample and we look into the genomes of the bacteria. And the genomes contain a lot of different genes. And one of the genes is really important for identifying bacteria. And that's the gene called the 16S Rna gene. That gene acts as a unique fingerprint for the different bacteria. And it's not just a simple fingerprint. So it works in that way that organisms which are more closely related to each other have more similar fingerprints. So even though we don't have a reference database with perfect fingerprints, we can actually say, what does this organism look like? And that's actually what we want to do when we try to infer microbes in a complex system. We want to look into these fingerprints and understand who are they belonging to. And it sounds very simple, but it's a more complicated process when you have a complex system. So the idea, what we're doing is that we first take a sample from our system, from the wastewater treatment plant. Then we do DNA extraction to get the DNA from all these organisms. And then we do some prepping in the laboratory to get it ready for sequencing. And then we sequence a lot of DNA from these organisms. And we do some bioinformatics on it. And what we end out with is a so-called amplicon sequence variant table. So that's parts of these 16S RNA genes from all the different organisms. And what we can see in an ASV table is the different samples we have analyzed and the relative abundance of the different ASVs or representatives of organisms. But we don't really get a name of the different organisms by doing that. So we need to do something more to get that. So what we also do is we do something called classification. So we take these small sequences of DNA and we compare it to a reference database where we have known names for the bacteria associated with these sequences. And then you can get some names for all of these ASVs. And when you combine that with the ASV table, you can get an overview of the community structure in these different samples. And in that way, get an idea about who are the organisms in our system. And that's really important. The problem here is that when we use traditional databases like silver and green genes, we get a lot of these red classifications. So that's where we can't really precisely say what organism is that. So we can say it probably on a family level, which would correspond to that we can identify an animal from the cat family, but it's really important whether it's a house cat or it's a tiger that we have identified in terms of what they can do. So we want to improve this. And the reason that we can't really go more into depth with these annotation or classification is that these reference databases here are incomplete, and they can be incomplete based on two reasons. So the first reason is that we don't really have the right reference sequence or the DNA sequence for that organism in our database. The other thing, which is also important, is that many of these sequences in our reference database don't have a taxonomy. That is a name for the different organisms. And if we don't have a name, we can't really talk about them. So we need to do something differently. So we need to create these better reference databases. And that's actually what the Midas project have been all about. So the Midas database is a database for activated slot and anaerobic digesters. And it started quite some years ago with the Midas 1 database, which were simply just taking this silver database, which is the common universal reference database being used. And then we manually curated the taxonomy for some of the important organisms in the wastewater treatment plant. So we figured out what were the abundant species in activated sludge in Danish wastewater treatment plants. And around these organisms, we tried to give names on this genus level for the organisms. And that was a manual process. We then introduced the Midas 2 a couple of years later, which then built upon a newer version of the silver database. And then we also expanded the organisms we were curating the taxonomy for. So we not only had the activated salt bacteria, but we also looked at organisms were coming into the wastewater treatment plants with the influent. And we also introduced anaerobic digests, which were located at these wastewater treatment plants to get a better overview of the whole wastewater treatment system as such. But these databases have some problems, which are, for example, we don't really do anything about these missing reference databases. We're only trying to curate the taxonomy of them, so only working with one of the problems. The other problem is that they are really difficult to maintain, so we have to go and look at a lot of phylogenetic trees and do a lot of manual creation. So updating one of these databases here takes a month work for one person to do. And it also only focuses on the most common taxa, or the most common organisms or bacteria in these systems here. So there'll be a lot of organisms we really don't learn news about. So we changed this with the Midas 3 database, where we, instead of using this universal silver database as a ground for it, then we created ecosystem-specific databases, which only contain sequences from wastewater treatment plants and anaerobic digesters. So Midas 3 was based on 1 million full-length 16S RNA sequences, or these reference sequences, and they originated from Danish wastewater treatment plants and anaerobic digesters, which were located at these wastewater treatment plants. And the reason that we could get a lot of these reference sequences that we needed were that we created a method which allowed us to do this full-length 16S RNA sequencing in high throughput. So we could get millions of these reads here. So in this paper here, we showed that we can actually create the same number of reference sequences, which were created in these university reference data by researchers all over the world for the last 20 years or so, just in a couple of weeks. So it really changed the technology. So now we can get all the reference sequences that we need for any ecosystem in practice if we can get DNA from those samples. The other thing we addressed was the lack of the taxonomy in the database. So we created a method to assign automated taxonomies to all of these organisms. So organisms didn't have real names in the silver database. We created artificial names for, which represented the same width as a normal taxonomic group. So we created artificial species which were at the same size as normal species, or had the same diversity. And in that way, we can actually refer to all the organisms which are in our system. So, of course, Midas 3 was just based on the Danish wastewater treatment plants and anaerobic digesters. We wanted to go a step further and create a database which were relevant for wastewater treatment plants and researchers all over the globe. So we expanded the Midas 3 database with 5 million of these full-length 16S RNA sequences from all over the globe. So we covered more than 700 wastewater treatment plants and more than 30 countries and on 6 continents. So we get a good overview of all the bacteria which are in these systems here. And of course, that has only been possible because we had a lot of good collaborators who could help us sample all of these wastewater treatment plants and ship the samples to us. So when we look into the diversity in these types of wastewater treatment plants, we can see that most of them use this conventional activated sludge process, but we also have a lot of other types of wastewater treatment plants. So we get a coverage of these types in our reference database too. And we also have different process types at the wastewater treatment plants. So we have similar wastewater treatment plants, which is indicated here by C. So that's wastewater treatment plants that only focus on removal of carbon. Then we have the ones which also have nitrification, some which in addition have denitrification, and then we have the biological phosphorus removal plants, which also try to recover phosphorus from them. So we have a broad diversity of different plants here, and also some spatial types which are not described here. So when we have these many reference sequences, of course, a lot of them would be from the same bacteria. So what we do is that we take these raw full-length 16S RNA sequences, and then we use them to infer exact biological sequences, which are called these ASVs. And we have around 90,000 of those in our Midas 4 database. We also right now working even further with the database. So we're making a Midas 5, where we are expanding the Midas 4 database with 1 million full-length sequences from anaerobic digesters. So in the Midas 4 database, we didn't include new digester samples. So they are only from the Midas 3, which were from the Danish wastewater treatment plant. So we want to have a database which covers also all the anaerobic digesters around the world. So that's how we expand this database, just by incorporating more of these reference sequences over time. So the one thing which is important is to see whether these ecosystem-specific reference databases are better than the universal reference databases, which are commonly used. So one way to evaluate that is based on, do we actually have the right reference sequences in our database? So what we usually do is we use some of these amplicon sequence data from community studies, which have been used just part of this 16S Rna gene. So there was recently a global diversity study in wastewater treatment plants carried out, and that data is based on wastewater treatment plants, which were not included in our study. So it's unbiased on the samples that we get. And we tried to take that data and then map it to the reference sequences we had, and we figured out how many had good references. That's references where more than 90% of the nucleotides in these reference sequences are matching up against the reference sequences. And what we can see here on this graph is how many of the different samples have these good references in different databases. So on the right, we have this Midas 4 database, which we created. And we can see that in this database, we have a much better coverage of these amplicon sequence variants compared to all the other databases. So this just means that we have the right reference sequences in our system. The other thing is about this taxonomy. So can we actually classify the bacteria more precisely? And when we say precisely, we want to go to what is called the genus level or the species level. So if we look at the genus level here, we can see that this additional taxonomy we created with this autotag procedures make us able to classify a lot more bacteria at the genus level here. So you can see with Midas 4, we are around 75% of all the amplicon sequence variants that we can actually classify at the genus level. Whereas with the universal reference databases here, where the best one is the silver database, we are around 30%. So it makes a huge difference in how precise we can actually characterize the bacteria. And with this Midas 4 database, we can even go to the species level. And it's not like you can classify all organisms at the species level, but around 45% can be classified using our reference database here. And that makes a huge difference, because with the common reference databases, we can't classify more or less any of them at the species level. So we get a completely new ability using this reference database in terms of how precise can we characterize the organisms. So one of the things we also did was to do a deep sequencing of all these different samples that we created using these traditional short read amplicon sequencing data. And that allowed us to figure out which were the most important bacteria in these wastewater treatment plants. So that's what we consider process critical taxa. And we defined 3 different ways of characterizing these core taxa. So we have something we call the strict core taxa, which are bacteria which are more than 0.1% abundant in more than 80% of the wastewater treatment plants. Then we have something called the general core, which is more than 0.1% abundant in more than 50% of wastewater treatment plants. And then we have conditional rare bacteria, which is more than 1% abundant in at least one wastewater treatment plant. And when we look into these systems here, we can actually see that when we characterize them and observe how much do they actually correspond to in terms of the total diversity within the system. So that's the number of total observed genera here. They make out a very small fraction in terms of numbers in the system. But when we go to relative abundance over here in the figure on the right, we can see that they actually describe more or less the largest fraction of the system here, more than 80% of the system in many cases. So it means that we can just look at these species here and actually learn a lot about the system. We don't have to care about the lower abundant ones for many cases. And we can also do this by the species level and see how many species we have. Here, these different core taxa makes out a very small fraction of the diversity, but again, makes out a large fraction of the relative abundance, closing into 50% of it. So again, by looking at a relatively few numbers of taxa, we can actually learn a lot. And to learn about why these, or see the importance of these de novo names that we create for the species, we can actually see how many of these genera we have observed in these different cores are de novo taxa. Then we can see that they actually make out a fairly large proportion of it. So for the general core, you can see it's 37%, which are without a name using traditional databases. And we can do the same for the de novo species, where more than half of them are not having real names. So we can only identify them based on our placeholder names created with this database. So what we really want to use this database for is to look into, for example, the global diversity of specific functional guilds. So for example, looking into the nitrifiers, which are generating nitrogen around in the wastewater treatment plant. So we have ammonia oxidizing bacteria. So we have traditional, the Nitrospira and the Nitrotoga who can do that. And we can see that it's mostly this Nitrospira which are in the system here, whereas Nitrotoga seems to be specific for certain countries. So it seems to be especially abundant in Europe here, whereas in Asia it's not that important, and in South America it's not important either. And then we have a nitride oxidizing bacteria here, where we can see it's actually only one genus which seems to be important. So it's this Nitrosomonas, which seems to be important, whereas Nitrospira, which is another one, doesn't seem to be anywhere except in a few cases in the UK and in Canada. So we can get this information about the bacteria here. But the really important thing is that we can now also go to this species level diversity and look what are the specific groups which are within these different genera, and see if they are specific for certain countries, or if there are more types which are important, or just a single one. So what we can see here is that for some of these genera, there seems to be quite a large diversity of different species which are represented in the wastewater treatment system. For example, like the top one here with the Nitrosomonas, it seems to be 10 types which are present here. And you can see I've written these remaining tags, which also incorporate 16 different species which were observed to some degree. So there's a huge diversity. But if we look into this Nitrospira, it seems to be dominated by one or two types in most plants. So there's a huge difference in how common the different ones are. And for Nitrospira, we have the traditional ammonia oxidizing or nitride oxidizing Nitrospira diffluvi, which seems to be in all plants, but in some of them we also have this Nitrospira nitrosa, which has been characterized as a common mox bacteria in some plants. So one which can also, in addition to nitrification, take on this ammonia oxidizing activity. We can't say it based on this kind of data here whether it is a common mox bacteria or not, but we can see that there is a potential for it being it. So when we have this data here, so I guess the take-home message is that we have this comprehensive reference database and taxonomy for the bacteria and archaean wastewater treatment plants. And we can go to this genus to species level resolution for the first time for the majority of the organisms. And also, I think a really important thing is that we have these 800 genera and approximately 1,500 species, which are important globally. So it means that we can actually, in theory, start to study those bacteria. And when we have done that within a reasonable time, we actually get a fairly good picture of everything which is going in these wastewater treatment plants. The important thing about this reference database is also that it can be used for cross-study amplicon investigations. So when you do amplicon sequencing, normally, then if you don't have the correct reference database, you can't really compare different studies. So you need to go to the taxonomies to be able to do that. And you can do that using this reference database here, which is also really important. So we get a language for discussing the importance of these bacteria. And what I also want to tell is that we try to collect all this information that we get from studying these organisms in wastewater treatment plants and anaerobic digesters, and collect it all in a field guide. So we have a field guide that you can log into, and then you can actually learn about each of the genera, what is known about this genera here, and whether we observe that specific genera. So finally, I would like to just discuss a little bit about what are the limitations of this 6NS RNA-based approaches. So one thing is the taxonomic resolution. So the question is this species to genus level resolution enough? Are the important traits for the wastewater treatment plants conserved at this level, or do we need to go to the strain level? If so, we need to use other technologies to go so far. And there are possibilities to do that. Then there's a question about something called primer bias. So when we amplify these amplicon sequence variants, we do not get a completely comprehensive picture of the microbial communities. There are certain groups of bacteria we are always missing using a specific primer set. So we need to take that into consideration when we analyze the data. There's also the question of that when we do this kind of analysis, we get relative abundance of the bacteria. So what would happen if the absolute numbers of bacteria change in the system? If we want to link it to process performance, it's really important about the certain number of bacteria rather than their relative abundance in a community. So we need to figure out ways to make or tackle this problem here also in the future. And that's also something we are working on. And then, of course, it's important that we provide a link between the taxonomies or the names of these bacteria and their functions. So many of these taxa which we identify are poorly described or not described at all. So we want to study more of them and put all this information into this field guide so we can share the knowledge, but many more studies are needed. So it's also what you call it, a call for the total community to supply us with data about this organism, so we don't have to do everything in our group, but can actually use the data coming from research groups all over the world. And then, of course, we want to implement these technologies here for process control. So if we want to do that, we need to consider the price and time to do that. And of course, the need of standardized protocols and intuitive software, which allows the operators at a wastewater treatment plants actually to do this kind of analysis. So you don't need to have a specialized researcher to do that. And of course, we need a lot of good examples of how we can actually apply microbial community profiling as process control and make a difference in that respect. So I think that was all I wanted to talk about here.
All right. Thank you, Morten, for this quite impressive overview of how you've been progressing with bacterial community reference databases. And I think with this last take-home messages, you point to the important message that we want to discuss today in the webinar. So as a listener, I would like to ask you, after having seen all these tremendous opportunities that reference database can offer us, try to think about, when you're looking at the current scheme, where you are at in the scheme. So if you are thinking about thinking about the fact that genomics might help you with solving the challenges that you are facing, or you're at the end of this chain, where you want to obtain or increase market share in applying genomics to all kind of problems, try to think about the challenges that you are facing, and perhaps you can share that in the chat with Ines, and then we could discuss this in the general discussion in the end. We also get some input from you in where in the chain people are with their challenges. The next, there might be a question from the audience, so I would like to give Ines some words.
Yeah, I have two technical questions for you, Morten. The first one is from Dirk Segas. He asked, how frequently is the Midas database updated, as well as the field guide?
So the field guide is being updated regularly or all the time. So whenever we get new articles out and people send articles to us, we try to update the database. But in principle, it's probably twice or 4 times a year we do it. So we try to collect all the information and do it. But in theory, we can do it all the way. The Midas database is being updated in principle when we get a new version of the Silver database, which is the reference database we use to create the background taxonomy on. That's the idea. And then, of course, when we do these major releases, like the upgrade to Midas 5, when we introduce the anaerobic digesters. Right now, we don't have any more plans than the Midas 5. So in principle, what will happen from there is that we update it when we get a new version of Silver to match that.
Okay, thanks. And then there's another question from Michiel Hootsmans. He asks how to decide that rare sequences are not important for the various steps in the treatment process. Is that based on the assumption that abundance is what matters? And then also note that primer bias may also influence the relative abundance data.
Yeah, that's of course right. So there could be many bacteria which are also in low abundance, which are important, specifically if you want to look into the degradation of pharmaceuticals, which you don't really need that many bacteria. You just need some which are specialized. So we don't really cover those in our approach. So we want to have the major degraders, and the importance of those are the flock formers, which can cause foaming problems in the wastewater treatment plants and those kinds. So in principle, we assume that there is a relationship between the abundance of a bacteria and its activity. So, but there could be many niches where that would not be a direct relationship in terms of abundance and so on. And of course, we always know that this primer bias. So what is really neat with the reference database is that we can actually evaluate the primer bias of different primers in silico. So we can see how good are different amplicons primers targeting the bacteria we see in our reference database, or whether we get a complete picture when we try to map these amplicon data. That's, I think, a feature that we can do with our reference database that you can't do in any other ways. But there's these biases that you need to be aware of.
Okay, then I have one last question for now, and that is that can the Midas also be used for other organisms, or is it only bacteria information that you have?
So right now it only covers bacteria and archaea. So we have included archaea also. So we had them from the Midas 3 survey where we sequenced a lot of bacteria and archaea. Then we also added reference data from the type strain of archaea to make sure that if you had a good coverage of that. So those are the two groups. So we can't look into eukaryotes at the moment, but it's something we want to do at a point, but we don't cover that using the same primers. So that would probably be another type of database.
All right, thanks, Morten, for these answers. I think it was quite clear, and they might provide the knowledge that the people need. I would suggest to move forward. So I would very briefly, next presentation, very briefly introduce myself. So I'm head of microbiology at Wetsus, and I'm a microbial ecologist. I will showcase some examples of how we apply genomics at Wetsus in various research themes. Can I get the presentation? Ah, thanks. That helps. All right. Thanks. I would like to show that the examples that I'm giving here are basically from this part of the chain. So we use the reference data, for instance, reference data that Morten just presented, to analyze our bioreactors and water samples to identify indicators and patterns that can tell us something about the biological processes in our systems. As a very brief overview, in how our research is organized at Wetsus, I give you some examples in the circles about subjects that are part of the research at Wetsus, and each of these subjects we apply genomics in a large part or a small part of those research projects. And they are basically colored by the different types of goals that we have to secure water supply, to monitor water quality, to recover resources from waste streams, or to simply reduce the use of water in our everyday lives. So I'm going to give you an overview of some of the examples in the 4 red circle subjects. And I will start with talking about an example from how we use genomics to monitor antibiotic resistance in our environmental waters. and especially around wastewater treatment. So I give you an example here about work that I'm involved with, and that's about carbapenem-resistant Enterobacteriaceae, and that means basically E. coli type of intestinal bacteria that are resistant to carbapenem-like antibiotics. And these antibiotics are basically already our last resort. So as soon as bacteria are going to be resistant against these kind of antibiotics, we really don't have a cure anymore if you get infected with these kind of bacteria. So these figures in this in the top left visualization show that in 2017 about 13,000 hospitalized patients were infected with these kind of bacteria globally, of which about 10% did not survive that, with major costs in the, in our healthcare system. So in a collaborative research project that Wetsus was involved with, the MEDUA project, it was investigated how antibiotic-resistant E. coli's were distributed along a river catchment, which you can see. It's the river catchment of the Vecht on the left bottom side, and the colored dots represent wastewater treatment plants that were putting their effluent waters after treatment into the river catchment. And we are interested in seeing what that effluent contributes to E. coli in the river. So, and part of that analysis, we were focusing on these carbapenem-resistant bacteria, and we found a couple in the influent of these wastewater treatment plants. And we selected about 200 of those to use genomics and characterize their complete genomes to see whether these bacteria are the same type of bacteria and whether they carry different mechanisms of antibiotic resistance, which you can see in the top right figure. There are many different genes involved that confer these bacteria with this resistance against carbapenem-like antibiotics. So while doing that, we characterized the full genome and compared all these 192 different E. coli's to each other, and then we found out that there was a lot of variation. So what you see is a grape tree, which looks like a bunch of grapes on a branch, and the closer they are together, the more similar they are genetically. So based on the genomic information. So what we found out is that in these different colors, so the different wastewater treatment plants along the river catchment, we found a huge diversity of Escherichia coli's genomes. But the question was, okay, we see different sites, we see different bacteria. At some sites, we see multiple different strains of these bacteria. But do they have these different antibiotic-resistant genes? Or do they all have the same pattern? And we have sampled these bacteria over a course of 2 years. Do the same bacteria pop up at certain times of the year across that river catchment? So if we look at this variation over time, and then we focus on the type of resistance genes that these bacteria carried, we can see on the x-axis the time period, and on the y-axis we see all the different treatment plants. And the colors indicate the type of resistance genes that these bacteria carried specifically against this carbapenem, so this biggest problem kind of case. And there we see that within certain wastewater treatment plants, across the 2 years, there were multiple different antibiotic resistance genes found, so multiple columns on the same line, and also particular types popped up at several times and at several moments. So these kind of information are important to guide our strategies on how to monitor antibiotic resistance genes at wastewater treatment plants. Sort of a mirror of what we have seen these days with monitoring COVID-19 in the wastewater treatment plants. So another example that I'd like to give is how genomics is applied to find bioindicators for toxic compounds in water. So the problem with toxic compounds, and this is the work of Antoine Carangera on the top left, the problem with these toxic compounds is that they are very low in concentration in our waters. They're very difficult to extract, and in many cases we don't know them even. So they might have some kind of molecular features which makes them toxic or genotoxic to us humans, but if we don't know what to measure, it's hard to keep track of them. So in a very elegant way, Antoine used worms, small invertebrates, to test how their genome, their gene expression reacts to certain and a wide array of different kinds of toxic compounds with different molecular features. And his idea was, okay, if they are exposed to certain toxic compounds, then their gene expression might tell us something about that. So he used these worms, and why worms? Worms are easy to cultivate. They have a very short life cycle. Their genome is remarkably similar to that of vertebrates. Well, you can replicate experiments quite easily with worms. So what he did is he took the worms in a petri dish, and he first exposed them to a whole range of different toxic compounds, and then tested as compared to control worms that did not have any exposure to toxic compounds, what kind of genes are expressed much more suddenly, likely as a result of that exposure to these compounds. And by selecting then those genes, so the ones that are red and orange in color, he took those genes and went further to develop information on whether the amount of these kind of compounds in water also linked to the amount of expression of those kind of genes. So what you can see here is an example of benzopyrene that he used, and a couple of 5 genes of which I made the name a bit easier to understand. And you see that in the cases of and the grayscale tells us something about the dose of toxicants that Antoine exposed these worms to. So what we can see here is that gene A and D didn't really respond to these to exposure of to benzopyrene, whereas the green ones did respond very strong. These genes are very nice indicators of expression to or exposure to benzopyrene in a dose-dependent manner. So the more you expose, the more genes there is expressed. So this kind of information is very useful. So what he did is he compiled many different genes that had these nice exposures and that could be used as bioindicators, and he put them on a chip. And with that chip, he could test a drop of water, basically, with an unknown a mixture of toxic compounds, and he could test how that would work in real life. So what he did, he took water samples of influent and of sewage from hospitals, nursing homes, and the general community, as well as influent. So those are samples 1 to 4, influent of the wastewater treatment plant. And he tested what the effect was of wastewater treatment on the toxicity of the water, basically, in a very broad sense. So he compared those samples with the samples that came out of the wastewater treatment and into the river, as well as surface water that was not related. And those are samples 5 to 8. So what he could clearly show is the very strong cleaning effect, basically, of the wastewater treatment on the gene expression of those worms for the genes that he selected. So in red you can see genes that were upregulated, those so basically went up because of toxicants, and in green the opposite was true. So that is a very from start to end kind of example of how you could use genomics to answer a question. Well, in the same kind of way, in these wastewater treatment plants, there is a lot of waste in the form of activated sludge. And in a next project, Angel and Rachel Pai, in their research team on biopolymers, they try to make use of that activated sludge, and they try to create biopolymers, or let bacteria in the activated sludge create biopolymers that could be used as a renewable and biodegradable way of making plastics. So what you see in these pictures is pictures of biomass, so bacteria in red, and the bioplastic kind of molecules that these bacteria make in green. And what they try to optimize in their system is the conditions in which bacteria make more of that bioplastic, of course. So they could transform up to, which you can see on the y-axis of this figure, you could up to 60% when these bacteria were also fed with calcium cations as compared to other cations that they apparently need to produce PHA, which are these biopolymers. So the question that then arises is, are these the same bacteria that are able to suddenly, under these conditions, produce more of those plastics, or are there different bacteria, which we can't see from those pictures, right? So we analyzed that these bacterial communities, so this biomass, and in this figure on the left, it might be a bit difficult to read, but what you should understand is that each colored symbol is one sample, that the two colors represent the calcium cation samples, and the orange color another ion, in this case potassium, and that the samples from the input activated sludge are still clustered together, so they were very similar in the microbial community. But then when they got calcium, they went to a different place than in this figure than the ones that got potassium, which means that the microbial community really changed. So the change in PHA production was really associated with a change in the bacterial community rather than their inherent ability to produce these PHA. So these shifts in the bacterial community you can already see in those colored bar graphs, where basically the dominant, the change in dominant bacteria change. So that's basically the relative contributions of these colors. Just as an example to understand how we could use genomics to understand the things that are going on in our systems that we don't have a good look at. Another example is the example of Suyas Gupta. She's working on in our sulfur theme, and they try to use waste streams, gas waste streams with a lot of sulfide, which are toxic, corrosive, and stink quite badly. They try to use those waste streams and to extract and recover sulfur from these waste streams, and they do that. They tested the bacterial communities in many different situations. So you can see that in the figure. There are different industries that have these kind of waste streams, and the bacterial communities differ a lot. But the main players always come back. So what they have tried, they take the bacterial community that comes from these industrial plants. And they put that in a bioreactor in Wageningen, and they try to optimize process operation, so turning all kind of buttons that enhances the production of pure sulfur, elemental sulfur, and they can get up to 95% of the sulfur waste that comes in to transform that biologically, so by the bacteria, into reusable sulfur. And well, that is basically what we can see here in this figure, where they put the inoculum from the industry plant and they twist the buttons in such a way that a couple of key players pop up. And these key players apparently are indicators of proper sulfur production. So what she then does is she takes this biomass and she wants to have easier to use indicators that we can measure fast, quick, and cheap, right? So what she does, she isolates those key bacteria. She isolates them on plates, and then she mapped the complete genome of these bacteria to figure out which genes are present that might tell us something about this sulfur conversion. For the future, that might be a very good way of understanding and easily monitor the biological activity in their sulfur systems. So what I hope with this presentation is to give you an overview of potential of genomics to understand a wide variety of systems and to answer a wide variety of questions. And I hope that by looking at this, you could also imagine how genomics could help you. So please hold that. And with that, I would like to go to the next part of our webinar and give the floor to Aleida. She is head of innovation at Orvion, and they apply genomics in real life. So the floor is yours, Aleida.
Thank you. Can I have the... Unless there's any pressing questions still.
No, not at the moment. So please send in your questions. We can also discuss them later. Your questions for Peter.
That's the one. Yeah, hi. So good afternoon, everybody. As already mentioned, my name is Aleida. And before I go into detail on how we apply genomics, let me just briefly introduce Orvion to you. Orvion is an environmental biotechnology company that was founded in 2013. And basically you could say we are microbial consultants within various environmental applications, such as bioremediation, water quality monitoring, water treatment. We have our own R&D facilities, mainly applied R&D, as well as some standard lab services that we can perform for our clients. The past few years, we've really been investing in BioT development. As already mentioned, it's important to be able to use and visualize all the data we're generating in an easy way for our clients who often have no microbial background. And in this way, we really provide a bridge between science and practice. So we're trying to get all the knowledge, or as much knowledge as possible, that's being produced in science and really getting it to be used in practice. So what kind of lab services do we have? We have a whole bunch of Qpcr analyses that we can perform for our clients just to quantify specific microorganisms or genes in an environmental sample. We perform NGS, so sequencing services, but also such things as lab-scale biodegradation tests or providing bioaugmentation material, so high concentrations of bacteria that you do want in your system. As I mentioned, a lot of focus the past few years on IT, so being able to design good Qpcr assays for the environmental sector, so not medical, but environmental. It's an important differentiation. And really turning all this data into information that can be used. One of our newest projects is called UDetect, which is a mobile Qpcr product that we developed that enables really non-specialists to perform Qpcr in the field and get results, if necessary, within 2 hours. So that's Orvion in brief, but of course you're probably more interested in to know what we do with genomics. And as I'm sure you know, genomics is used in research and in the medical sector as well, I think increasingly a lot already. So I could say that it's already a routine method that's used in these sectors. However, in our sector, it's possibly still a bit further away from being a mainstream method. But still we use genomics, mainly shotgun sequencing, to answer some typical questions that we get from our clients. And clients could be water boards, looking at water quality or at wastewater treatment, drinking water companies looking at drinking water treatment or how to keep it stable, and also the bioremediation sector. So that's really degrading certain contaminants in groundwater. So that's a typical sort of array of our clients. And what kind of questions do we get? Sometimes it's really easy. It's who's there, so which bacteria or microorganisms are in my sample. That's I suppose, relatively easy. We sequence them and we give them an interactive file that shows them to a genus level who's there. But then often, of course, the next question is, are these good bacteria? Are they bad bacteria? So, for example, if there's a certain precipitate being formed, which organisms might be doing that? Could they be pathogens, or the smelly ones, the ones that are producing sulfides? Then we're really looking more at who they are and how we can maybe get rid of them. Sometimes, of course, they're also the good ones, so the ones that are degrading a certain contaminant and the ones that you want to have in your system. After that, it can get a little bit more challenging sometimes, because then, of course, the question is, okay, if I do have these bad guys, or I want to keep these good guys in my system, how do I have to change my system in order to get rid of them or keep them in there? Of course, that's another way we will use genomics to monitor it in time, or to monitor how different additions of chemicals or different process conditions might affect the microbial composition in a positive or in a negative way. And then I think the most challenging one, where we're really looking to apply genomics in practice, is how does this knowledge allow me to improve and control my microbial system? So that's sort of the holy grail, I suppose, that after having done all the previous, answered all the previous questions, that you can go back to your client and say, if you turn this knob a bit to the right, or monitor this once every week and it stays under this threshold, then you're fine. So that's sort of a little bit how we work in this area for our clients. And I want to give you just a couple of practical examples. I don't think I have time to go in detail, but if there are any questions, then let me know. Just to give you an idea of what we're doing and the type of questions that we get in practice. So the first example I wanted to give is a project we did for a water board, Waterschap de Dommel. And they have a wastewater treatment plant with a constructed wetland after it to treat the effluent coming out of the wastewater treatment plant. And they put this constructed wetland here because the wastewater treatment plant is discharging the water into quite a sensitive stream which is ecologically quite sensitive. And of course, having a constructed wetland means there's extra hours and people necessary to make sure that it's working correctly. You have to make sure that the water stays open and not too many plants grow. So it's an added cost, basically, for the water board. And what they wanted to show was that it has this added value. So it's worth this extra investment because you're actually treating your effluent to a higher degree and getting rid of even more chemicals, but also microorganisms that might impact the receiving water. So the main question for us was, what's the effect of this constructed wetland in removing certain microorganisms from the effluent? And can we then see the discharge in the microbial composition of the receiving water? So we took samples from various spots in the constructed wetland, and of course upstream and downstream from the discharge site. I really can't go into all details, but just to show you some of the results we got. First question was, is the wastewater treatment plant microbiome, and also of course the sewage microbiome, is it being removed efficiently? So on the left bar chart, you see the data that we generated, which has been filtered for fecal indicator bacteria, or just the really well-known fecal microorganisms that were in the effluent of the wastewater treatment plant. So this is the water that's usually discharged into the surface water. And what you can very quickly see is that in the discharge water, so they have two discharge points, the northern and the southern discharge point, both being treated in the same way, that yes, a great degree of these fecal indicator organisms are being removed efficiently. So that's a really good result, because you don't want these microorganisms in your surface water. We also, of course, confirmed these results by performing a Qpcr, which is a quantitative method, because, of course, this is all relative abundance, but you want to know if your absolute numbers are also decreasing. And there we saw the same trend when looking at certain indicators. Another thing that was quite interesting, as you can see, the first one is the southern discharge point seemed to have a slightly lower reduction rate than the northern discharge point. And it was actually noted while they were taking all these samples that there was a shortcut circuit in this part of the constructed wetland, in the southern part. So some of the water wasn't going through the whole constructed wetland. And we saw this in a lot of the results, that the removal efficiency of this constructed wetland was, in fact, a lot lower than the northern side. So this also helps to demonstrate how important it is to upkeep the constructed wetland and make sure everything's working correctly. Because if it goes through everything, in the end, you remove a lot more. Another interesting thing that came out of this is that certain species wasn't being removed very efficiently by the wastewater treatment plant. So the genus of the Qpcr, and there was a publication from the same group as Morten actually, that had also seen this, that wastewater treatment plants don't seem to be very good at removing this genus. And it's a genus you want to remove because A, it's a potential pathogen, and also for microbial resistance, it's one you don't want to have in your surface water. And here we also could demonstrate that, hey, through this post-treatment in the constructed wetland, you were actually removing more of this Qpcr. And once again, you can also see that the southern constructed wetland was performing a little less than the northern one. So this just helps basically our clients to go to their investors and their managers to say that this extra investment is worth it because in the end, we're treating the water better. Another example I wanted to show you how we're really applying these genomics techniques in practice is for the bioremediation of groundwater contaminants. This is another field that we do a lot in, and once again, it's the microorganisms doing the work in the groundwater, degrading these contaminants that are available. And there's a lot of sort of physical chemical parameters that are monitored and measured to see if degradation of these compounds is occurring. But one of the missing links, as always, was the microbial community. Are the organisms actually there that are able to degrade these contaminants? And if we try to stimulate them, do we actually see them increasing when compared to a reference where there is no contamination? And this is actually something we do quite regularly. We call it now the genetic plume profiling package that we do, whereby we analyze a number of groundwater samples using metagenomics. We include all the process parameters we have, so the redox conditions, the concentrations of the contaminant, and compare it to a reference to see if it looks like there's a biological activity, or the potential anyway, to degrade these contaminants in site. And it's something we do a lot. This is just one of the examples of a site contaminated with naphthalene. And you can see very quickly that this is already filtered for species that are relevant to degradation of naphthalene, which is a whole bunch of different organisms. And you can very quickly see that this Desulfitobacterium is the dominant species in this contaminated site, and especially when compared to a reference. Our advice then was also to, of course, monitor this microorganism, specifically with Qpcr, which we now do, and to stimulate this site to enable these organisms to become more active. And based, among others, on these results, a pilot has been started at this location to stimulate degradation by adding sulfate in one pilot, and the other pilot nitrate, to see if we can really enhance the microbial degradation at this site. So it's a very specific way in which we quite quickly can get a lot of data on the microbial composition at these sites, but really help the contractors and also the people sort of determining what has to happen at this site, to see if this microbial degradation is a feasible option, and if it's not, if we can then stimulate it maybe to get degradation happening, but also to monitor using Qpcr the organisms that we found to be of most importance at the site. So it's a very practical way of using genomics in practice. Then the last example, I think, that I wanted to show you is a project that we're performing once again for waterboards, where we're also looking, like Morten already described, at the activated sludge in these wastewater treatment plants. And what we're really trying to do here is to get all the information, so the microbial information, all the process data, which, for example, is like the middle, all these graphs of all the different process data that they're collecting every day or every minute or every second, as well as all the bio-IT that we perform, and put it all really into one platform, which we've called now the Activated Sludge Platform. And this wasn't really to answer a specific question, but to help water boards manage all the information they have, but also to help them include all this microbial information that is being generated all over the world, and use it in their daily practice, which is something that they find very difficult because they still think it's all very complicated and very far off from their daily practice. So in this dashboard, what we have is various visualizations. I won't go into all detail, but here on the left you can just see the taxonomic, the heat maps of various taxonomies on genus level. But you can also put it on different taxonomic levels if you want to go a bit higher. As well as on the right, you get these bar charts. So every bar is a different sample, and the combination of different genera gives you different colors. You can filter this data. So we have some predetermined filters, for example, for filamentous organisms or for nitrifiers or for organisms that are involved in phosphate removal. And if you want, you can also include the process data, so temperature or concentrations of various parameters that they're measuring. And in this way, you enable the person basically to browse through their own data and look for correlations. I don't have much time left. Excellent. I'll hurry up a bit. Another thing we've included is the possibility to look at correlations, so very sort of basic correlations between different parameters that have been included in the databases to see if there's any obvious trends occurring. And this is just one example here on the right of a certain organism that apparently likes warmer temperatures, because on the top is the temperature that's being monitored. So this has a correlation that occurs, and of course you need to look further into seeing if this is very significant or not. And where we really want to go with this dashboard is eventually also for the operators to be able to make certain threshold values of parameters that are important for them to determine if their treatment plant is working correctly. So do you have enough nitrifiers for nitrification, or are these filamentous bacteria increasing so much that it might start to cause problems for your treatment plant? So that's sort of the next step that in collaboration we're working to really make it more practical for the operators that are there every day determining how the treatment plant is going. So these are just a few of the examples I wanted to show you of how we're using these genomics in practice. But as always, there's some challenges that we come across, and these are just a few that I think are very relevant to the clients as we work with. One being the turnaround time. Usually they want to know yesterday what was happening and not sort of, we might have results for you in a few weeks' time. No, they need to know now what to do, because you need to know now if you need to change some of the process parameters or stop your drinking water production system or goodness knows what. Another main, great challenge that we come across is the complexity, not only of the method, going from a sample to the DNA to the results coming out, but also the type of results we're presenting. So for you guys, I'm sure it's all very easy to follow, but for somebody who doesn't really know what a bacteria is, it gets very confusing very quickly. But also, of course, microbiology in general tends to be slightly less predictable than all the physical chemical analyses that are performed. If the temperature is this, then it's going to be that for a while, and you don't need to worry about it too much. Whereas with microbiology, it can change quite quickly. Then another thing really that I think is, the question if genomics is going to be mainstream is other sort of upcoming techniques or some existing techniques that are more suitable for sort of this routine monitoring. I already mentioned Qpcr, of course, which is a cheap and easy and quantitative way to monitor systems, but also things like metaproteomics, which is sort of the next step of not only who's there, but what are they actually doing, which is, of course, a question that we often want to have answered. And the last thing is a sort of the nice to know versus the need to know. I mean, we present our data and it's always, oh, this is so interesting and fascinating, but yeah, now what? You know, what do I do with it? What does it mean to me? Versus the need to know, which often is, legislation says no E. coli is allowed to be in my drinking water, so that's all I really need to know, and all the rest, yeah, nice, but I don't really need to know about it. Or for a water board, it's the effluent shouldn't contain ammonium above this concentration, or shouldn't contain nitrates above this concentration. So yeah, all of the other stuff is nice, but yeah, I don't really need to know it. So that's a big challenge, to find these examples of how you can really make it need to know versus nice to know. In short, can I still go through this? Yeah.
Sure.
Go quickly. As far as I think we're all concerned, is that microbial data is invaluable for the water sector. And there are a lot of applications that we can think of in which it really has a lot of added value. The challenge really to unlock the data in a way that it can be used, not only for one application, but for a lot of different applications. You want the same pipelines, the same methods, and the same results as much as possible. The nice thing is they're being used more and more often. I mean, just as our company, the number of analyses we perform is going up and up and up. And the added value is really recognized. But it's not mainstream yet, and the question is, and I wonder how you guys feel about that, will they ever be mainstream? That, I suppose, is the main question. So remains me to thank you for your attention, and thank you very much for the invitation to come and speak here today.
Thank you, Aleida, for this very nice overview of the possibilities in the field of applying genomics. A very short opportunity, no questions directly, then perhaps people could still add that to the chat and we could discuss that later in the final discussion. Because then I would like to already give the floor to Adrie Atsma, a project leader at Vitens, and as I already mentioned, also Dutch delegate at ISO. Please take the floor.
Well, thank you for the introduction. Small overview about Vitens. We are the drinking water company, and I'm working at the laboratory where we use standard procedures to see how the water quality is, especially from a microbiology perspective. And yeah, we use standards, and those are standardized. And also the government is looking at us. Hey, you must use standards because every drinking water company has to use the same method to give a reliable result about how the water quality is from a microbiology perspective. Before I start this presentation, I thought, how can I give you a simple overview about standardization and why will it support innovation, but also the other way around. Innovation will also help us for standardization. And I used some metaphors. In March, when the lockdown was over, the first thing that I said to my wife, I want to go to a Greek restaurant. So, I was very surprising. I'm very keen on moussaka, especially the bechamel sauce. And I was a little bit brutal to ask the waiter, hey, do you have the recipe of the bechamel sauce? And then the waiter said, it's no problem. You have to use these kind of ingredients. 1 spoon of salt, 2 spoons of oregano, and so on and so on. And so I took it in my head. I think, okay, so I can also take the challenge. I will also reproduce it also at my home. But in the kitchen, they use very sophisticated materials, and my interpretation of a spoon was a little bit different that from the Greek restaurants, because I took a tablespoon, for example. I used an old instruments. I was not that precise. I thought, 200 mils of milk. Yeah, perhaps 300 is also okay. So I was making the bechamel sauce. The lower part was okay, but the bechamel sauce was awful. So the conclusion was, you can see it. It was a bit pale, and the taste was very awful. And my son was also complaining about his stomach when he ate that. So it was not a very, very good example of taking a simple recipe from a Greek restaurant. if I wrote this down and I was looking at the kitchen what kind of materials they were using, yeah, I think it would be wise to do that for the second time when I make the bechamel sauce. Another way, I had to standardize a little bit, and that's what more or less a metaphor for it. What is actually standardization? Yeah, it's meaning, yeah, you have to write things down. You must make agreements with companies for you to say, okay, this is the best way to do it, and also scientists like you guys to say, hey, this is the best way to perform the method to get reliable results. So you have to write these things down, and to avoid disagreements between companies, but also between scientists, and also yeah, a special part, avoid incidents, because it can also be that you get a very risky method, and if you do that perform well at certain steps, yeah, incidents can happen. But there are different types of standards. I'm more in the ISO and the CEN and the NEN standards at the moment. Those are the more the national, the international ones. But actually, when you are at the laboratory, you have also your own standard operational procedure where everybody is going to use that. You make as an organization, you say, okay, we're going to use this standard operational procedure. That's actually also a standard. And you can also make a national one. And if you want to make it official, then you have to use the national standards from the NEN. And if you want to go international, because we're also working a lot together with the European Union, and they say, hey, we have to work together, you can make an ISO standard for that, so the whole world going to use your method. And the funny thing is that Aleida and I are also making a standard about how to work with Qpcr, and yeah, that's also quite a challenging project, because I go further on the next slide about that. I like to give another metaphor. Now, the connection between innovation and the standardization. Yeah, it's a difficult one, because we need innovation to develop new methods and to create new standards. Yeah, we also do a lot of work with research institutes, and I like to show you a small metaphor about Simon. This is his name is Frank, and he wants to do a PhD study at Wetsus, for example, and he has an assignment from Vitens to develop a next generation sequencing method, where he is going to screen on messenger RNA markers which are involved with the denitrification process. Perhaps a new project for you guys, but. Now, but what is Vitens? Yeah, Vitens is actually we at the moment we have a lot of automation, because we get a lot of samples, and if we want to use complex methods, that we have to automate it, and also a lot of other companies are using a lot of automation. Actually, it must be quite simple, and but we want to have a fast method, like Aleida already mentioned, to get a fast result, because we produce water, the customer drinks it, and yeah, if something's going wrong, yeah, you don't have to wait 3 or 4 days for it before you get the result. But for us, the most important ones is yeah, we must have a reliable method. So that's why we are also using that a certain kind of standards. So yeah, go back to the metaphor. Frank is assigning his project. He's doing a lot of literary study. He's developed a method with NGS, where it's for example, an RT-PCR method. He is going to test some samples from the environment. We get them samples from certain treatment plants. And yeah, he gets nice results. He presents his results to the business, and he writes some publications about it. We're quite happy. And his end result is actually that he found 3 markers based on NGS data, and which are involved with nitrification processes. So and he said, hey, we can make also a quick, fast method for us. So there's also already an important message that I want to give you. Keep it simple and keep it fast if you want to introduce a new method in our lab. So like most publications, you get quite a large discussion about certain types of topics. And Frank had developed 3 genetic markers, but are they selective enough? Can you rely on it that these markers are unique for the denitrification process? So actually you have to validate it, and there are certain standards for that. What does it do when you get also other metals involved? For example, iron manganese, will it involve the whole process, and also and he get also some genetic markers which he did not expect to get, and his results were not really reliable, but there was enough thing to build on, to do more research on. And on table, he showed also some results, and he did not predict. He said, I got those 3 markers, and I did also a certain kind of reproduction experiment. And he took day 1, he found, for example, at the first marker 10,000 copies, and the next day he get only 1,000, and the third day he gets 10. But he didn't store for example, the genetic material well. So that's also quite things that you have to put inside your standard operational procedure. For example, and this is a small example that you have at your lab. So how do you deal with storage of genetic material? And I'll go to the next slide. So actually, when you're starting an experiment, you have always get in your mind, we're going to do some research and development, and we have to have, if I'm going to do this, is this a method that it can be quite simple? And so there are also laboratories like VTENS can use it and also automate it. And of course, there are a lot of complex things on it, but if you get a method which says to the company, now you can predict what the nitrification process will be, and it's as fast, then it's also very interesting for us. And it's not that we say, okay, we have to immediately standardize it like ISO, for example, to get it high. But if we, for example, get a method and we're going to test it at the laboratory, we're going to promote it and to say, hey, we have a nice method there where you can also, for example, interesting for another drinking water company. And they will also try it out, and after that, if it looks promising, you can standardize that, and normally it goes to an N level. And if it's also interesting for international partners for us, you can standardize it by ISO. So, but that's a long way. It's, for example, your development starts now, and you have a method. It can already take 10 years before you get an ISO standard for that, for example, for a new method. So standards meets innovation, and it's also for you guys at the laboratory who are doing a lot of research work. Look behind you. Is your instrument that you are using? That's also a critical question from us. The instrument that you're using for example, pipettes or PCR systems, are they calibrated or are they checked? And you have also to validate your results and use also internal controls, but also controls to see if the processes that you are doing during your experiments are they going well, and so that you can say, okay, the results are reliable. And you can also validate already the new methods, especially when you're going to method development. Please perform some validation to see if, for example, the markers are specific for that process, or and is that also suitable for what kind of type of water that you want to use it? For example, drinking water has less of inhibitors, but if you want to use it for surface water, they have a lot of inhibitors, so the test will not suitable for that matrix. So yeah, innovation helps also in standardization, because we need also insights from you guys to see how we do a certain kind of trick, and we see that it gets a better quality of DNA, for example, or we have a certain an alternative fecal indicator for you, which can better predict the fecal pollution instead of E. coli and Enterococci. Yeah, that starts with innovation. It starts with we doing something, and yeah, that's quite important that we. Yeah, it's not only the direction we're going to standardize. We have to tell you guys how to perform the method. No, we need also the opposite. From hey, you're going to invent new things, and we're going to implement it at our lab. But keep in mind, it must be simple. It must be also be certain kind of automated. The possibility to automate the method. So, yeah, there's also some useful standards that I want to promote. Is that there are already a lot of standards for PCR, and I've read 2 days ago there was also published a standard for next generation sequencing already, especially about data treatment. I'd like to mention that we are developing a new method how to work with PCR in the international level. And at the national level, we have also already almost published a practical document, the 7394, how to work with PCR. Yeah, I want to recommend it also to read that document. So this is more or less what I want to tell you about standardization. Yeah, if you got any questions and if you got any ideas about standardization, but also can also help you, please contact me. And I would also thank you, my colleague from the NEN, Laura Mott, who was also supporting this presentation. So this was for me what I want to tell you.
Thank you, Adri, for this clear overview of how you work with standardized methods at your lab, but also how we could translate our own work and perhaps the work of the audience into working in a standardized fashion, and that there is not a direct necessity to scale it up to this ISO level to work in a standardized way. Ines, are there any technical questions for Adri?
At the moment, not for Adri, but in the meantime, a question came in from Tessa Pronk of KWR related to the presentation that you gave, Peter. And maybe that links to this one, because she asks, how well verified is the test for toxicity, so the test from Antoine? Can it already be used for this purpose? And here again, this standardization and automation has been a part of that project, right?
Well, perhaps you might be the one to answer that question as being the supervisor of Antoine.
Yeah. So I'm personally involved in this project, and I can say that standardization or making it suitable for routine use has been also part of the challenge for Antoine, and also resulted in the preparation of a manuscript now. Also in view of that, you want to transfer this in time to for routine use to a company or a lab like VTens. And we're not there yet, because now we're in this phase, Adri explained that you want to test it more in practice and see if it withstands all these questions. So we're still looking for people and companies that are interested to go the next step. But I can say that in the line of a EU project, we are going to continue on this. So hopefully in the future we can also show that it also for routine use is a suitable bioassay.
Yeah, that sounds promising. That is good new news, I would say. I don't know whether it completely answers the question, but at least there is some perspective that these answers will be generated at some point in time.
Yeah, I think it relates to what Adri is asking. Please, at the moment you're already doing the innovation, think about this. We may not reach that because for innovation you also need a kind of a freedom. But you have to keep in mind that in the end, the results that you obtain you can be able to translate to practice. And you have to keep that in mind. I think that was your suggestion.
Clear message. Yeah.
Okay.
Are there any more discussion points, more general discussion points that you want to bring in, Ines?
Well, there are no more points from the audience at this moment. But the question that I had starting out thinking about this webinar is, how can we make it easier to come from this innovation to this application? We've seen 4 presentations, each taking a part of this challenge, and but what can we do to make this a more smooth transfer? So maybe the presenters, or maybe Morten, can explain what he thinks is a way forward.
Yeah, that's a good starting point, perhaps.
Yeah.
So, being really in this applied field, Aleida and Adri might have already some ideas about this. Could you, Aleida, share your thoughts if you have them?
Yeah. I suppose the main question starts with which questions are you trying to answer? So is it, you have the questions from the innovation perspective, which I think you and Morten very much managed to demonstrate. I think those are doing really well already. I think there's a lot of application in that respect. I think the main clue really to get it applied more and more in practice is a collaboration. You need this collaboration between the end users, being the water boards, the drinking water companies, the early adapters, which I suppose is more the Orvions of this world that are trying to take all this knowledge and start using it in practice, and getting the standardization, because as soon as, or standardization may be legislation, I'm not quite sure exactly how to walk through that, but as soon as you get to this point that it's a need to know and not only a nice to know. So as soon as the water board starts saying, okay, you're not allowed to discharge so much antibiotics resistant genes into the surface water, as soon as that becomes legislation or something that's more tangible for them to require to monitor, then it's going to happen. So I think that's, and it's difficult. It's definitely not something that's going to happen very quickly, because as Adri already mentioned, it takes a while. I think I found that the most, the shocking thing of joining in with this whole standardization discussion is how long it takes, how many opinions there are, how you need to try and find this consensus, then get it on paper in a way that everybody agrees, then get it internationally. I mean, it takes years to get that far. So it's definitely something that takes a while. So I think, off the top of my head, those would be a number of the points that would start looking at a more routine application of genomics, or the knowledge that comes out of the genomics that's being used.
Do you want to tag in? Do you have a different view on that, Adri? Or
No, it's
is
more
it
or less the same same thought that I had. At Fitens, it's more the question. There are common questions from the business, and one of the examples that we try to find out is we already use a method for I think more or less 100 years for detecting of E. coli. But if we find E. coli, is that a real big problem? Yes or no? So we also try to find out with next generation sequencing. Hey, is there something going on? And can we find also an alternative for that? Because we know already with the knowledge that we had that are perhaps better fecal indicators that we can get. But the main question is coming from Fitens, is it important? Can we get as fast as soon as possible a result, but also in the perspective of what's legislated. So we have to find a fecal pollution. It must be 1 E. coli per 100 mil. Can we get that? And that's the starting question before you're going into it. But on the other hand, if you doing innovation, the discoveries are most you do it by coincidence. so I don't want to stop the innovation in a way. We have to explore, but you have to keep in mind what is the main question what the drinking water company wants.
Yeah, so that links pretty nicely to the need to have a nice to have discussion, which is basically counterintuitive from an innovation standpoint, right? But from that same, in that same kind of area, Morten, I was wondering how you look at standardization of methods, whereas you're continually, and I mean, it's inherent to your project, this continuous growth of reference data, and molding that in a way that is useful for sort of a standardized analysis, but which which changes every time you would basically rerun it year after year. What would be your vision on standardization from your perspective?
Well, it's very important that we do a lot of standardization to actually be able to use our data for anything. So when we do these short read amplicon data that we use for surveys, we have really strict guidelines for how we create the amplicons, which primers we use, which conditions we use, and also how we do the DNA extraction and so we can actually compare that data across each other. And of course, as you mentioned, these reference databases, it's also important that we can get the same name for the same bacteria every time we do it. So that has been something we worked a lot on with these placeholder names for the bacteria. So when we update the database, we make sure that the way we have created these artificial names, they stick when we also add more sequences in the future. And that's really important for being able to actually transfer the knowledge we have from one version of the database to the next one. So I think that's really important. And also when it comes to how we should apply these methods that we create, I think it's really important to have this idea that we want to start with a specific goal from the industry. So we need to figure out what is the research question we want to address, or the mission of this methods, because what we can then do is we can actually look at the possibilities in terms of the technologies we have to figure out the best solution for that question. Right. And to do that, we need insight into the system. For example, if we want to look into what could be indicator genes for a specific purpose, we need to know something about the diversity of these genes within the systems to make sure that when we create primers, that they actually target the specific microbes and don't have other a non-target that it can also hit and so on. Yeah. So I think that's really important that we try to combine what we do in academia also with the industry and have collaborations to tackle specific problems or tasks.
Yeah, so this integration of end user versus innovation, there seems to be some kind of barrier that needs to be broken to make, to basically bridge this gap. Also, Adri pointed to that direction. At Wetsus, we try with how the organization is organized to let the questions from the field flow into the research society to tackle these kind of problems. But I think for applied genomics, is what I hear from you is that we need to do that in a more general sense, being able to find each other to direct our innovation processes, and not just for discovery, but also for quick and fast application. Yeah, I think that in this way we nicely identify these kind of problems. Ines, I don't know if there are any more questions, and perhaps no. Well, with that, I would like to close this webinar with thanking you for your input. I really enjoyed the way we could integrate each other's angles into the same kind of chain of events that we need to get applied genomics more to a standard way of working in the challenges we face. I hope that the audience has also gotten a good idea of how it might fit into their challenges. And if you have ideas, specific or unspecific, do not hesitate to contact us or through Wetsus or directly to either one of us. And with that, I would like to thank you and wish you a very nice evening.